Meet Avery, a scientist who prepares drug candidates for clinical trials. Like everyone else, she depends on drug distribution models developed over 50 years ago. With each project, Avery has concerns about the conclusions drawn from these biased models. Her worries are valid, since assumptions made by the well-stirred model have failed her in the past. If Avery and her team could get a precise picture of the drug's journey through the animal at the cellular level, this would solve her problem. She would improve her odds for clinical success and avoid watching years of work and millions of dollars slip down the drain. Fortunately, she can, with tissue modeling based on MALDI imaging. By switching to tissue-based models, Avery's team moves to the next pillar of drug discovery, a label-free technique that maps drugs, metabolites, and endogenous molecules within tissues, all of which has streamlined candidate evaluation at pharma companies worldwide. To make this powerful technique applicable to all of pharma, Bruker proudly introduces MALDI-2 on the timsTOF fleX. Here's how it works. The typical MALDI event uses a laser, localizing molecules to a small pixel on the tissue surface. MALDI-2 adds an orthogonal laser pulse, ionizing even more material, providing enhanced sensitivity and chemical range. Now all types of compounds, including statins, sterols and steroids, NSAIDs and many others, plus their metabolites, can be mapped against pathological studies. MALDI imaging on the timsTOF fleX with MALDI-2 is a complete solution. Included are easy-to-use workflows, optimized software and hardware, and consumables for each step of sample preparation and analysis. Avery knows that the difference between the two approaches is dramatic, sort of like trying to pinpoint a single raspberry in a blended, well-stirred smoothie versus finding one in a fruit bowl, to determine exactly where it is and who its neighbors are. timsTOF fleX tissue imaging with MALDI-2 provides the same advantages, and as a result, Avery's team substantially improves their models of safety and efficacy. Now, Avery's time course studies show causality from a biological perspective, finally adding necessary molecular component context to histopathology and the disease state. With this new pillar, detailed drug location is an integral part of the drug development discussion. Additionally, Avery's group can also map mechanisms and metabolic pathways, showing true PK-PD relationships, which has no parallel in the pharma industry. Easy-to-use MALDI-2 on the timsTOF fleX for molecular imaging is essential for Avery's team and their success. How will you use this groundbreaking technology to advance your work?